This video is just going to be a quick overview on some thoughts on muscle. Obviously, for those who are studying the muscular system more in depth, I have a lot of videos which go through this in greater detail. Muscles, let's just pick one, the biceps brachii, actually a little bit more complex than most people imagine. Actually, technically, muscles are organs since they have different types of tissue present. In addition to muscle tissue, they have neurons, which are sending commands, but there's also connective tissue wrappings. And so the fact that there's multiple tissues present, each muscle, like the biceps, is technically an organ. Now, there are actually three different kinds of muscle tissue in the body. So here you can see skeletal muscle. The cells are enormous. I mean, they can be wide enough to see without a microscope. They can be, so long they extend past the ends of a microscopic side. They can even be measured in feet, as some of them. They have these brilliant bands, these striations. They can be so big because during embryonic development, hundreds of little embryonic cells can come together and fuse to make these long tubes, which are then, not cells per se, but now the fusion of lots of cells. And that's why there can be hundreds of nuclei resulting from these hundreds of cells which have fused. Skeletal muscle cells can heal a little bit, not all that well. And they are voluntary. So if you wonder whether you have skeletal muscle at a place in your body, the easiest way is to try to move it. If it moves, that means that you have skeletal muscle there. It is the only type of muscle which is under voluntary control. Cardiac muscle is found only one place in the body. It is found in the heart. It also has striations, but the cells aren't as big as skeletal muscle. They didn't result from the fusion of lots of embryonic cells. So there's only one centrally located nucleus rather than hundreds. One of the obvious ways of identifying cardiac muscle are these bands known as intercalated discs. So because the heart generates a great deal of pressure, there are two types of junctions found where cardiac muscle cells meet, which strengthen cardiac muscle and allow electrical messages and other things to pass from one cell directly into another. Smooth muscle is a third kind of muscle. It lacks striations, but it is found in many parts of the body. It lines most of the digestive tract, most of the respiratory tract, the urinary and reproductive tracts. It lines blood vessels. It's what we see in the pupil of our eyes, controlling the constriction there, or allows our hairs to stand on end. It is called smooth because it is not striated, and it is involuntary, as is cardiac muscle. So both of these can receive commands, say from the nervous system. We can control the contractions of smooth muscle and cardiac muscle, just not voluntarily. So muscle cells are unique in a number of ways. They can contract, they can be stretched, which is good because when the biceps brachii is contracting, obviously we want the triceps to be stretching. They can conduct electrical messages known as action potentials. So both the cells of the nervous system and muscle cells can have electrical message stretch over them that is a unique feature in the body. There are collagen fibers from connective tissue around muscle cells, around groups of muscle cells. And this then means that when the muscles contract, then they pull on these collagen fibers. And if the collagen fibers then attach to a bone, say in a tendon, now the muscle contraction can actually then move the bone along with it. So here you see collagen fibers. Once again, some collagen fibers will wrap around the muscles, but then these could form this band of a tendon, which then insert to a specific spot on a bone. And so when this muscle contracts, then it pulls the bone along with it. Here you can see the striations of a muscle cell, but here you can see the fibroblasts and the collagen of the tendon that they then produce. If one wanted to describe what muscles do, then at some point one has to talk about these structures known as sarcomeres. These are small things which are called the functional unit of muscle. That's because a muscle, like say the biceps, is made of muscle cells, and muscle cells then are made of these tubes called myofibrils, and myofibrils are then made of sarcomeres. So, essentially a big muscle like the biceps is lots and lots of sarcomeres arranged into cells. And so if one can understand what happens in a sarcomere when muscles contract, then one could explain the contraction of the whole muscle, because the sarcomere is the functional unit of the muscle. Essentially, a muscle is lots and lots of sarcomeres. So there's a sarcomere, there's a sarcomere, there's a sarcomere, there's a sarcomere. The sarcomeres make the myofibrils make the muscle cells, and then the muscle cells compose the much larger muscle. And so once again, when one studies muscle, at some point one studies these sarcomeres. And just, very, very quickly, there are two major proteins in the sarcomeres. There are the actin filaments, which attach to the end of the sarcomeres. And then there are the myosin filaments in the middle. And this muscle fiber then has a cell membrane which can send electrical messages down to the sarcomeres. So an electrical message can go over the sarcolemma, that's the excitable cell membrane, and then go down these tubes, these tubules, so that electrical messages can reach the sarcomeres where we tell them to contract. Because one of the proteins, actin, is attached to the ends of the sarcomeres. These are known as Z-discs. Then if we could simply bring these closer together, then the sarcomeres would contract, and then the whole muscle would contract. And so the secret to a contracting muscle is the protein in the middle, which pulls on these actin filaments. So if actin is in blue, then myosin is here in red. And the myosin proteins pull on the actin and then bring those sarcomeres closer together. So that will make one sarcomere shorter. And if we can do that in all of the sarcomeres, then since muscle cells are essentially made of lots and lots of sarcomeres, as the sarcomeres then contract, then this will shorten the sarcomeres and thus shorten the muscle cell. And so when a muscle is contracting, these myosin proteins here in red, they have these globular heads which are reaching up, grabbing the actin and pulling on them. There's a little bit more involved, and my videos go through that. But the last thing that I will say here about that is we obviously want to control this. So at rest, that doesn't happen. So your muscles aren't flailing around at random, because at rest, the blue actin molecules, they are covered by a rope-like muscle called tropomyosin and another molecule called troponin, which holds it in place. So at rest, muscles aren't contracting, because at rest, the spots of actin which get pulled on, they are covered by tropomyosin. Troponin holds it in place. And then when your nervous system sends commands, calcium is released. The calcium is stored in this place called sarcoplasmic reticulum. So here in pink, when calcium is released, that binds to troponin, moves tropomyosin out of the way, and now the myosin molecules can access the actin and then pull on them. Okay, and so the contraction of muscle is then controlled by the interaction of all of these various proteins. And their interaction is often referred to as the sliding filament theory, because these protein filaments can then be observed as they move across each other as myosin grabs onto actin and pulls. Okay, and so this is the secret to muscle contraction. Muscle is made of sarcomeres, and in a sarcomere, the actin is tied to the ends, the Z discs, and the muscle and the protein that's in the middle, myosin, will attach and pull on the actin, which brings it closer to the middle of the sarcomere. And this is controlled because at rest, the active sites of actin are covered by other molecules, which prevent myosin from pulling on. All of this then is controlled by the nervous system. And we could talk of the excitation, contraction, coupling, because nerves touch muscle cells. And so when electrical messages travel down nerve cells, and so here you can see nerve cells, these are nerve cells which are touching muscle cells. The electrical messages of nerve cells then get converted to electrical messages on muscle cells. And then those electrical messages travel over the cell membrane through those T tubules to where the calcium is released. And then calcium will start the contraction of the sarcomere. And so thus the electrical messages of nerve cells can become the electrical messages that travel over muscle cells, which then result in contraction. Now, obviously, there's a lot of other topics which can be of interest, especially as we start talking about, muscle contractions and exercise. And here we start getting into the energy sources which are used. So that's a bit beyond what some of my students would study unless they were, say, going into allied health programs. So I have videos if it's of interest, because, for example, if you were going to do a short bout of intense exercise, like weightlifting or running a 100-yard dash, first we would use up the ATP stores which are in muscle cells. We would generate a little bit more because our muscle cells have creatine phosphate, which would then generate more ATP. But all of this would be exhausted within a couple of seconds, or within a short period of seconds. And so then we would resort to our third source of energy, glycolysis. If we were running a very short race, like once again, a 100-yard dash, we would simply take this sugar and break it into ATP. The advantage is that we would certainly get energy that way, and that would keep us going in exercise. But the problem is that lactic acid would build up, which would cause fatigue. And so if we were doing glycolysis and stopping at that point, our muscles would get sore. We might, at some point develop, cramps. And so we would need to stop. If you were going to do a much longer bout of exercise, so if you were going to go jogging, for example, you couldn't do glycolysis and stop. You would then have to undergo what's called mitochondrial respiration using oxygen. So when you're jogging, you're running slower than when you're sprinting because you need time to get oxygen to the mitochondria to generate more ATP. This would generate carbon dioxide, which could then be released as a waste. Now, there's a lot involved in exercise. So for example, when lactic acid builds up, you can briefly send it to the liver, and your liver can package it again to make more glucose. This is why sometimes when you're running, you might develop, a stitch in your side because the lactic acid going to the liver can disturb its homeostasis. And once again, there's a difference between the types of exercise which are more anaerobic. So we're not using oxygen primarily because it's just occurring too fast. We're breaking molecules down to get ATP for energy, but we fatigue quickly. We get cramps, etc. As opposed to the more aerobic respiration, where we have time to get oxygen to our mitochondria, where we can break pyruvate down completely into carbon dioxide. And so thus we're not building up lactic acid, and we can continue exercising for longer periods of time. Um, and so, the types of exercise that we do, whether we fatigue or not, the energy sources which we use, these and other topics, are often, studied as one studies the muscular system. And I have videos on these if they are of interest.